In 1988, neurologist Robert Katzman identified a clinical anomaly that has frustrated Alzheimer's research ever since. A substantial minority of elderly patients die with severe amyloid plaque burdens, enough to warrant a definitive Alzheimer's diagnosis, yet they maintain entirely intact cognition to the end of their lives. This observed fact challenges the assumption that the physical accumulation of amyloid plaques universally drives cognitive decline. Clearly, the presence of the pathology alone does not guarantee the destruction of the mind. This diagram outlines the central paradox dividing neuroscience today. One body of evidence suggests losing microglial homeostasis, when immune cells leave their resting state, initiates synaptic collapse. Opposing research indicates this exact activation builds a protective barrier around plaques, preserving cognition. Microglial activation cannot logically be both the root cause of dementia and the exact mechanism of cognitive resilience. These researchers are describing the same biological process from opposite sides of a conceptual wall. They are missing a layer of the brain's defense architecture. Our inability to predict dementia from plaque counts reveals a structural blind spot in our current biological model. The unified model of Alzheimer's progression relies on 4 established layers: the microglial homeostatic state, the protective perineuronal matrix around neurons, synaptic function, and the propagation of tau proteins. To resolve the microglial paradox, we must integrate a 5th physical layer, the containment substrate. Containment is a physical work product, an executed cellular mantle built by microglia that wraps an amyloid deposit and compacts it into a dense core. A pathologist measuring bulk sees the same plaque load in both brains, but the tissue itself reveals a physical contrast. One brain leaves its deposits naked and sprawling, while the other holds them within tight, compacted envelopes. Biological resilience emerges from this architecture, the physical integrity of the wall shielding the neural network. In its homeostatic state, a microglion is highly active, constantly extending and retracting branches to survey its territory and clear minor debris. Microglial activation is a licensed excursion, a departure from homeostasis to wall a plaque. The TGF-beta and SMAD signaling loop is the chemical tether pulling the cell back once containment is finished. In the first failure mode, insufficiency, the cell is too starved to make the departure, leaving the plaque unmantled. In the second, dysregulation, it deploys, but the return path decays, trapping the cell in chronic inflammation. The 2 conflicting theories in neuroscience are simply describing these 2 different failure modes. The resilient brain successfully governs both halves of the excursion. The barrier building program requires heavy machinery, highly destructive proteolytic enzymes, like matrix metalloproteases and phthepsins, used to clear debris and remodel tissue. In the lower half of this map, we see these enzymes destroying the perineuronal nets, the protective matrix shielding the brain's inhibitory neurons, which directly triggers cognitive decline. In the upper half, those identical enzymes act protectively. They build the tight microglial mantle around the plaque. Restraint is spatial targeting, confining destructive enzymes strictly to the plaque's address, rather than spraying the surrounding tissue. The destruction of the brain's protective matrix occurs when a disciplined gardener loses its spatial address and sprays its tools into the open brain. This physical cellular mantle directly controls the concentration of the sprawling, toxic protofibrillar halo, preventing it from leaking into the surrounding brain tissue. If the mantle is breached, escaped particles drift into the neuropil. These tags, C1q and C3 complement proteins, stick to nearby healthy synapses. This tricks the immune system into reactivating a pruning program, commanding it to eat its own healthy neural connections. Neurites exposed to the unchecked halo swell into dystrophic axonal spheroids, which are heavily loaded with misfolded tau protein. Dystrophic neurites act as the primary seed reservoir for the spread of tau across the brain's connectome. A successfully contained plaque starves the downstream networks of the chemical tags that order synapse destruction and the tau seeds that spread the disease. The traditional model of resilience assumes a brain with perfectly pristine, undamaged systems. Resilience is more accurately modeled as a quantitative threshold, a running ledger comparing the advancing pathological load against the sum of the brain's biological restraints. Real brains experience partial failure continuously. A local matrix thins. Plaques remain unmantled. The value of the restraint side is always fluctuating, and the brain compensates. Dementia occurs the mathematical moment the total sum of parallel restraints falls below what the pathology demands. This explains why therapies targeting a single isolated layer consistently fail. Once the total threshold is crossed, the interconnecting systems drag each other down. Cognitive decline is sequential. The clinical event of dementia is the moment the brain's running total of biological defenses finally falls short of the bill. We can apply this architecture to the recent clinical failures of TREM2 agonists, drugs designed to aggressively activate microglia to clear pathology. A TREM2 agonist acts purely as a departure signal, forcing the microglia out of homeostasis to build the barrier. Because patients' native return signals have often decayed by the time they show symptoms, pushing the cells out without a way to bring them back strands them at the plaque. This forces them into a state of chronic dysregulation. Administering a departure signal alone is iatrogenic. It converts an insufficient immune response into a violently dysregulated one. The rational intervention requires two distinct chemical signals given simultaneously. We must administer a containment enhancer to push the gardener out to the plaque, paired with a homeostatic restorer to repave the road home. Mashing the immune system's accelerator fails to treat the underlying disease. Effective pharmacological restraint requires us to manage both halves of the excursion, demanding the work and guaranteeing the return.